Hi, and welcome to my OCRA A-Level Biology revision session with me, Christine. So today's lesson, I want to look at genetic diagrams, specifically monogenic inheritance. So let's just remind ourselves about variation. So population of a species will show variation. You will see that if you look around you right now, if you're in a place where there are more people. Now, because it's within a species, we call this intra-specific variation, because we're looking at the differences within, for example, the species Homo sapiens. Now, variation basically means the differences in observable characteristics, those phenotypes, those physical traits that we can see. And that is to do with your genetic material that you possess, as well as the environment for which you live in. So these are two factors that cause variation. Now we're going to focus today's session on the genetic material of an organism. So you should know that a gene can have different forms, alternative forms, and that's what we call an allele. So if there is two or more alternative forms of a gene, that is what's known as an allele. So if we look at, for example, seed color in a plant could be green or yellow. Therefore, the gene for seed color has a very specific position on a chromosome. Now, we can see that these are linear chromosomes, so therefore we know we're talking about eukaryotic organisms. We can see that the gene, a section of the DNA, is located at a specific point. That's known as the gene loci. Now, the next thing we need to understand is that because we're saying we have different versions, alternative forms, we can have what's known as a dominant allele that we represent with a capital letter. Now, that dominant allele, that sequence of DNA bases, is always going to be expressed. And what we mean by being expressed is that it is actively influencing the organism's characteristic. So if we have a dominant allele, we represent it with that capital letter. If we have a recessive allele, we would use a corresponding small letter. So in this case, I've got green and yellow as my seed color. Therefore, I've got a capital G and a small g because it's the recessive allele is the corresponding small letter for that dominant allele. And in the exam, they will tell you either which one's dominant, which one's recessive, or they'll indicate that with using the genotype. Now, do be aware that as you can see with my petal color, I have pink and purple. Well, both of them start with P. So I have to take more care when I'm looking at what the recessive trait or what the dominant trait will actually be. So the key thing to note then, what causes variation? Well, one of the things to cause variation is a gene mutation. A permanent change to the genetic composition of an individual is going to cause variation. So for example, instead of it being a thymine, we have a nitrogenous base of cytosine, an incorrect copy. Now, if that incorrect copy is not identified and repaired during the cell cycle, that could mean that during DNA replication, there is a permanent change, a gene mutation, a permanent change to that genetic composition. So when we look at the alleles and we know that we have the dominant allele, we've got the recessive allele. If we have two alleles which are the same, they are called homozygous alleles. So here you can see I have two which are green and two which are yellow when we look at the seed color. So the genotype, the combination of alleles, would be for the ones on the left, capital G, the phenotype, the observable characteristic, would be green seeds. If we're looking at the petal color, we've got capital P. Therefore, our phenotype would be pink. Now, they are homozygous alleles because the alleles are the same. So that would be homozygous dominant, or I can have homozygous recessive, small g, yellow, small p, purple. So it's important you know the difference between the genotype and the phenotype. If it's the same, they are homozygous alleles. If they are different, they are called heterozygous alleles. And you need to remember, if it is heterozygous, you only need to have one of the dominant alleles for that to be expressed. So therefore, if I have capital G, lowercase g, I will have a phenotype which is a green seed. If I have capital P, lowercase p, I will have a phenotype which is a pink petal. So it's important we note the difference between whether it's homozygous or whether it's heterozygous, and we also understand whether it's a dominant or recessive allele that's passed on. So Gregor Mendel studied the inheritance of different characteristics in pea plants, and what he did is he looked at tall pea plants and short pea plants, and when he crossed them together, what happened was he identified that all of the work that he got gave him the tall plants. So all of the F1 generation, the first generation that he bred, were tall plants. Well, his work was before all other scientists who actually discovered chromosomes. So he just looked at observable characteristics and started to know that all of them were tall. Well, because of that, he would therefore be able to identify now, because we know about dominant and recessive alleles, that the genotype for tall has to have the dominant allele, and the genotype for short has to have the corresponding small letter. So therefore, because we know that tall is dominant, we would use the capital T to represent tall, and the short would have to be the lowercase. Now, through meiotic division, we know that we are going to reduce the number of chromosomes to produce gametes. So therefore, by meiosis, we would end up with gametes that only have one of the possible alleles. Now, when we're looking at homozygous individuals, it doesn't matter which gamete we look at, because obviously they will both contain exactly the same. So what happens if we take two F1 generations and we then cross-pollinate them? What would happen? Well, what we would do is we would see that they would actually have potentially two different gametes. So a capital T or a lowercase t. When that haploid gamete is going to be produced through meiosis, that would then come together through reproduction and fertilization. We would end up with a possible combination, which would be TT, capital T tall, capital T lowercase t, also tall, because remember that dominant allele, capital T lowercase tall, and two lowercase t's would be short. So therefore, our phenotypic ratio, our observable characteristics, would be a ratio of 3 tall to 1 short. Now, obviously, we wouldn't just do it once, we'd do it many times over. So we would look at the number of plants that we observed, and we would hopefully end up with a 3 to 1 ratio. So that's how we do a monohybrid cross. But how do we know that a tall plant is homozygous dominant or heterozygous? What can we do to identify that? Well, what we would do is we do what's known as a test cross. A test cross is where we're going to take our tall phenotypic plant and cross it with a homozygous recessive. Now, if the tall plant were heterozygous, when we cross it with a short homozygous recessive plant, our ratio changes. Instead of us having a 3 to 1 ratio like we had with heterozygous and heterozygous, what we get now is a 1 to 1 ratio when we take a tall heterozygous plant and we cross it with a short homozygous recessive plant. So if we get 1 to 1 tall to short, rather than all of them being tall, that indicates that our tall plant had to be heterozygous for that allele. So some genes actually have more than 2 versions. So they don't just have the dominant and recessive, they can actually have multiple alleles. So if we look at our blood groups, this is a prime example where multiple alleles can be found for a gene. So you can either be blood group A, blood group B, blood group O, or blood group AB. Well, how is that possible? Well, that's because codominant alleles, when you have a pair of alleles that are both shown in the phenotype, like we have with blood group AB, that means that allele A and allele B are both dominant. Therefore, we say that they are codominant. So you could have a blood group of being A, and therefore you would have either the genotype IA, or you could be heterozygous for it, IAO. If you are blood group B, same scenario, you can be homozygous dominant, or you could be heterozygous. If you are blood group O, you are homozygous recessive. So therefore, O is the recessive allele. A and B are as dominant as each other. They show codominance. Therefore, if they come together, if someone inherits allele A and allele B, they therefore will have the phenotype of blood group AB. So how does that work? Well, if we took someone who has blood group A and we took someone who has blood group B, and they therefore were heterozygous for their trait, that therefore would mean that there's a possible gamete which would have an A allele and a gamete which would have an O allele. So if we took those and we cross them together, what you would get is all 4 possible combinations. Now remember, the phenotypic ratio is a 1:1:1:1 ratio, but it may be that the parents produce offsprings that all have exactly the same blood group. That is because it is by random fertilization of which egg and which sperm are fertilized. Now, I am actually blood group O, my mum is blood group A, and my dad is blood group B. Therefore, I know that because I'm O, I have the recessive alleles. I know that both my mum and my dad have to be heterozygous for me to have inherited the allele for blood group O. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. And if you haven't already done so, please do check out my revision platform, www.aiqchat.com.